In the wake of the escalating global COVID-19 pandemic, the need for rapid development in vaccinology has never been greater. When dealing with a pandemic like COVID-19, vaccine development and discovery of therapeutics is essential to improving outcomes. The next two case studies address the metabolomics applications and Metabolon's capabilities, which are uniquely positioned to address the need for actionable insights in vaccine development. Further, to provide a comprehensive view for more informed decisions to drive operational and quality improvements. This case study talks about how metabolomics can be used to identify an ideal media composition for vaccine production optimization. The objective of this study was to elucidate virus-mediated changes in host cells and interrogate the pathways and mechanisms involved in infectious bursal disease virus replication. Infectious bursal disease is one of the most economically important diseases that affects commercially produced chickens worldwide. And although largely controlled by vaccination, new virulent strains of the virus still remain a threat to the poultry population. The challenges, as are found with any bioprocessing approach, are upscaling production of the vaccine or biologic or metabolite once it's developed. Scaling up vaccine production is not necessarily a linear process. In this study design, DF1 cells, which are a continuous chicken embryonic fibroblast cell line, were analyzed across a time course, either in an uninfected or infected state. Comparing the infected versus uninfected resulted in profound alterations in a number of different nodes of metabolism. First, in central carbon metabolism, looking at glycolysis and the TCA cycle, it appears that glycolytic intermediates were increased, whereas TCA cycle intermediates were decreased. Additionally, an adjacent pathway off of glycolysis, the pentose phosphate pathway, saw increased carbon input from the glycolytic pathway. Now, the importance of this is that the pentose phosphate pathway, one of its main roles, is to generate substrates or precursors for nucleotide biosynthesis. Now, if you imagine when a virus enters a host cell, it basically hijacks the machinery to try and replicate itself and infect the host. And a virus, imagine what it's made up of. In this case, in IBDV, it's an RNA genome similar to COVID-19, and it's encapsulated by a protein. And when it replicates, it needs to transport from cell to cell, so it needs vesicles, which are made up of lipids. And in this case study, we saw increases in a number of amino acids, which make the protein for the capsid protein. We saw increases in a number of purine and pyrimidine metabolites, the nucleosides and nucleotides, which would be needed for generating and replicating that RNA genome. We also saw increases in a number of lipid components, which would be needed for vesicle formation and transportation of the virus within the host cell. Taking what we learned from the metabolomics and marrying that with a design of experiment methodology, the researchers were able to come up with a strategy to formulate a new optimized media. The options for optimization could have included things like glucose or lipids, amino acids, nucleotides. Because glucose uptake limitations were apparent and lipids are not necessarily the most bioavailable metabolite in cell culture, the researchers focused on amino acids and nucleosides. So applying this to the mathematical methodology of DOE strategy, the researchers developed a model which gave them actionable opportunities for an empirical test of their media. And again, based on the metabolomics data and the DOE strategy, they were able to generate and optimize a media which was able to give them a 20-fold increase in titer. Now, if you imagine, a 20-fold increase in titer saves not only money, but time, which is of the utmost importance in the present COVID-19 pandemic. The lessons learned from this example, which is from a chicken virus, hold true for human infectious diseases and will no doubt play a crucial role in the understanding of human viruses and vaccine development, particularly in respect to COVID-19. In addition, Metabolon has experience in vaccinology and the effects of virus in other viruses such as influenza, herpes, rabies, in addition to infectious diseases caused by bacteria such as anthrax and tuberculosis. This case study talks about identifying a limiting nutrient in order to optimize a platform process. The challenge in this one was the need for an optimized production process. Comparing it to the needs of today in vaccine production, the ideology really holds true. In this case, it was a large pharma working on the optimization of a platform-fed batch process for a therapeutic monoclonal antibody using a GS-CHO system. Similarly, in present-day situations, using a cell line for virus replication in a COVID background, the ideology of limiting nutrients, which could be supplemented to optimize a process, the same ideology is there. The Metabolon insight that was generated from this came from analyzing both cell pellets and spent media. The global Metabolomics library that was used to generate the large amount of data could be distilled down to a number of key observations. The first being markers of membrane leakage. In the media, we identified metabolites such as glucose 6-phosphate, which should not be found in the media in a cell culture background. When glucose is phosphorylated in a cell, it's typically retained extracellularly. So when we see it outside the cell, while it's supposed to be maintained intracellularly, it's an indication of membrane leakage or unhealthy cells. In addition, we found a number of metabolites which were found to be depleted or exhausted over time, choline being one of them. Choline is a very crucial phospholipid head group component, a precursor for phospholipid biosynthesis. And in a bioprocess cell culture, where you're asking a cell to replicate as fast as possible and produce a protein which must be transported, which requires more lipids, choline depletion can have a drastically negative impact. Based on the metabolomics and the recommendation to the researchers to supplement the media with choline, they were able to increase their monoclonal antibody titer. Not only that, the percentage of aggregates was found to be reduced, which is an indication of improved quality. Optimizing this media and product yield allowed for saving time and money. As mentioned before, the lesson learned from this example of a monoclonal antibody can easily be transferred and relatable to virus replication in bioprocesses in vaccinology applications for any virus, particularly relevant to present-day COVID-19 pandemic.